All right. Well, good afternoon, everyone. My name is Dr. Tracy Newman. I am a pediatrician, associate professor of practice in public health, and the medical director at North Dakota State University Center for Immunization Research and Education, also known as CIRI. Today, I have the pleasure of introducing Dr. Dan Wilson, who will be presenting this webinar titled New Technology, Old Fears, a walk through the science and myths of mRNA vaccines. Dr. Dan Wilson is a molecular biologist who earned his doctorate in biological sciences at Carnegie Mellon University. He currently works as a senior associate scientist at a biotech company and spends part of his free time exercising his passion for science communication on his YouTube channel, Debunk the Funk with Dr. Wilson. His channel focuses on debunking anti-vaccine myths and disinformation using sources from the primary scientific literature. This webinar is being recorded. A publicly accessible recording will be available on the NDSU CIRI YouTube page following the presentation. For those seeking 1 credit of CME or 1 MOC point for the American Board of Pediatrics, please follow the link available in the webinar chat to take the pretest. We are excited to be offering pharmacy credit for this webinar. Instructions for redeeming CPE will be available at the end of the presentation. If there is time, Dr. Wilson will answer questions at the end of the webinar, so please type your questions during the presentation into the Q&A feature of the Zoom platform. If you prefer to email them anonymously, please email ndsu.cire@ndsu.edu. Dr. Wilson, you can begin.
Thanks for that introduction, Tracy, and thank you all for joining. I'm very thankful for the NDSU team for inviting me to give this talk, and I think it's an important topic to discuss in these times. So hopefully you all learned something and we can talk about some cool science. So with that said, let's get started. Okay, just really quick, I have nothing to disclose. And let's get started. So misinformation flourished during the time of COVID, and it's important to understand why that happened. So COVID was a situation that was pretty unprecedented. We had times where we were home for weeks, if not months. There was a lot of upheaval in the regular workspace. There were a lot of changes to daily norms. And of course, there was also a virus that was spreading and killing at most about 4,000 Americans a day at one point. And that was all very scary and very, very big in everyone's daily life. And so when there are these kinds of unprecedented changes and big events happening, that creates a lot of genuine fear. And that genuine fear can turn into kind of a desire to look for easy answers or look for easy way outs of complicated situations. And so during those times, misinformation, which does often provide easy, simple answers as opposed to the more nuanced and complex realities that we face, can be a more approachable or more palatable thing for people to accept during those times. And so during COVID, there were a lot of organized anti-vaccine campaigns that sprung up, gained steam, and took up a lot of space online. And it helped a lot of people feel better about the events that were going on. And we see this in the data. We see that COVID-19 and vaccine-related myths have kind of gained steam and gained in numbers. Even as the years go on, some of them have gotten more and more popular or kind of fluctuated in popularity. So ideas that COVID vaccines change people's DNA has gone up in since 2022, and a lot of these other myths have kind of either stayed the same or not changed very much. So it's important to address these myths and continue to educate so that these myths don't continue to grow. So that brings us to how do we address this misinformation? Thankfully, there is research on how best to address these kinds of myths, how to have these conversations with people. And of course, you have to be willing to have conversations with people who might be misinformed or might disagree with you. Uh, but in order to have those conversations effectively, we have to do things like familiarize yourself with false claims. If you're in a conversation with somebody and they make a claim like COVID vaccines change your DNA, I read this paper, and you've never read that paper before, you don't know about the claim it's talking about, it might be harder to have that conversation. But uh, you can, if you have the facts on hand, you can quickly address those topics and talk through that claim with whoever you're having the conversation with. But either way, we see that research does show that follow-up is really important. So whether you know what the person is talking about in the conversation right then and there, or not, it's important to follow up and have other conversations. Like at the end of a conversation, say, I'll look at this paper if you read this article, or I'll watch this video if you listen to this scientist, and can we come back and talk some other time? So building trust and see these people to see that, you know, us scientists and medical professionals are real people, and we uh, do our best to put out good work, and that these facts aren't really nebulous, that you can really talk through them and teach them. That matters and is important to a lot of people. So, objectives for this talk is we're going to discuss the science of how mRNA vaccines work and how they're made, so that we have this kind of base understanding of the basic science, so that we can go into conversations with a good amount of knowledge and be able to communicate that during these kinds of conversations. We'll also explore the breakthroughs that made mRNA vaccines possible. There's at least one breakthrough that was really, really important that often uh, leads to some misunderstanding in how they work. So we're going to discuss that. And then we're going to identify some specific common myths and think through them using the background that is discussed in this talk. So one thing that is really important, and what we'll start with, is just explaining how vaccines work. This was a really big, I think, sticking point for people during the pandemic. People had a kind of an expectation on how vaccines are supposed to work. And in reality, the vaccine didn't meet that expectation. But vaccines don't really work by preventing infections. And I think a lot of people expected COVID vaccines to prevent infections, prevent them from getting a positive test, prevent them from getting symptoms. But vaccines prevent disease. And this has been true since vaccines have been around. They they work to prime your immune system to recognize a foreign pathogen. And when your immune system is recognizing that foreign pathogen or pieces of that pathogen in the vaccine, it builds an immune memory. And that immune memory is what really is going to matter in preventing the disease, not the infection. And this image here just shows a schematic of what a typical immune response might look like, where a naive B cell, a B cell is a cell that creates antibodies, encounters a pathogen or a foreign antigen, and it divides, it clonally expands, it produces a lot of antibodies, and then those levels of antibodies will go down. And then in a secondary immune response, you get another peak and then another decrease and plateau. So it's this plateau period where you can still encounter infections and even experience symptoms, but it's this peak, this memory response that is going to clear the pathogen before symptoms or the actual disease happens, I should say. So you might get symptoms, but it might not be like severe symptoms. And a good example of this is polio vaccines. But we're going to talk a little bit more about that in this next slide, where we explain why COVID seemed to be different. Why is it that someone could get a polio vaccine and not get polio their whole life, but they get COVID vaccines and then they test positive for COVID? Is it the vaccine? Is it something else? What's going on? It turns out it really comes down to the pathogen's incubation period. So in this table here, for example, we look at polio. Poliomyelitis from the time the virus infects your cells to the time you get paralysis or other severe symptoms is 5 to 20 days. And during that time, your body has enough time to have an immune memory response for those B cells that initially encountered the vaccine to clonally expand, to build antibodies, and for T cells also to get mobilized around your body to address the virus and clear it before it causes something like paralysis. So you could get infected with polio and never know it because you don't get paralyzed and you don't get those severe symptoms. However, if we look further up in the chart with diseases or viruses like influenza or the common cold and coronavirus would be in the same family of the viruses that cause common cold, those incubation periods are on the order of 1 to a few days. That's not fast and that's not long enough for your immune memory response to kick in and clear the virus before you get any symptoms. So that is why we see a lot of people still testing positive for SARS-CoV-2 even after they've been vaccinated for COVID. But the vaccine is also why we are no longer in lockdowns, why we are no longer seeing thousands of Americans dying every single day from COVID. The vaccines give us an immune memory response so that we can address the virus and clear it before it gets to your lower lungs and causes severe respiratory distress. So that's how vaccines are supposed to work. They're supposed to prevent disease, not infection. This was not communicated super well during the pandemic, and I think it's an important point to go in to know going into these conversations because a lot of people still might think that the vaccines didn't work because they tested positive after getting vaccinated. Another thing important to remember about how vaccines work is just to explain the components of a vaccine. Um, Vaccines always will contain either a weakened form of the pathogen it is vaccinating against, a killed pathogen, or just pieces of the pathogen. Immune cells are going to see these foreign proteins, these foreign parts or pieces of the pathogen, and build their immune memory response against those foreign pathogens. And this immune memory response is going to be composed of B cells, which again produce antibodies, but also T cells, which function differently. And the T cells are also really important to remember, but we'll tuck that away for a few slides later. So antibodies bind epitopes, which are parts of a pathogen, essentially. So pathogen has foreign proteins, and an epitope is a part of that foreign protein. Whereas T cells will bind peptides or short sequences of proteins on their MHC molecules. Both memory types of these cells will persist after a vaccination and be ready to engage a foreign pathogen when those foreign proteins show themselves. So all vaccines accomplish essentially this goal. Show foreign proteins to your immune system, have your immune system respond, and build an immune memory response. This has always been how vaccines have worked, and now we have lots of different kinds of vaccines today. From the oldest vaccine around, like a live or inactive or weakened vaccine, to all the way to nucleic acid vaccines like mRNA and DNA vaccines. They all work on these same principles. And so when someone is talking about fears they have about mRNA vaccines, you can explain these kinds of basic principles to get the point across that mRNA vaccines really do function at the base level exactly like every other vaccine that we've had for decades or centuries. And these are the details of how mRNA vaccines work. You have an mRNA vaccine with a piece of genetic material. And that genetic material is, of course, the mRNA. And that mRNA is encapsulated with lipid nanoparticles, which is essentially just fat. So you can just tell people it's just nucleic acids, which we all have, and fats, which we all have. And the mRNA in the lipid nanoparticle is going to encode for a piece of the pathogen. In this case, it's coding for a piece of SARS-CoV-2, the spike protein. It codes for the spike protein because the spike protein is on the outside of the of the virus. It's the first protein that your immune system is going to see, and neutralizing it with antibodies is going to help prevent the virus from spreading from cell to cell. So once the mRNA vaccine is injected into someone's shoulder, then the lipid nanoparticle is going to essentially fuse with a cell. So the fats of the lipid nanoparticle are going to fuse with the fats of the cell membrane and deliver the payload, which is the mRNA, into the inside of the cell. And once the mRNA is inside the cell, ribosomes, which are the machines in your cells that read mRNA and produce protein, are going to do their job. They're going to make protein, and that protein is going to be, of course, a spike protein, which your immune system will then see and build an immune response towards. So let's pause and recap. If a common concern is that COVID vaccines don't work because you still get COVID, you can easily address this with facts by explaining that SARS-CoV-2 is a short incubation period virus. It's not going to be quickly neutralized by your immune memory response if your vaccine or if your last vaccine or last exposure to a virus was a few months ago. So you might still get those symptoms as the virus is replicating in your upper respiratory tract, but your immune memory response is going to kick in and clear the virus or stop it from reaching your lower respiratory tract, where you would typically get those severe symptoms like being hypoxic or having respiratory distress, which would be normally what would land you in the hospital. Another question that or a common concern that people might have is, why should they get vaccinated if they can still spread the virus? And the answer to this is that vaccination actually does reduce the risk of your transmission, because you might be infectious for SARS-CoV-2 for several days after you first get infected. But if an immune memory response kicks in and starts clearing the virus during that time, you can reduce that window of infectious shedding, thus reducing your chances of spreading it to someone else. However, it doesn't eliminate it. That's why SARS-CoV-2 is still around. We're not going to get rid of it. It's here to stay. So that's a big reason to still get vaccinated if the concern is, what good is it doing anybody else? But you should also just say that, I want you to be healthy. I want you, person who is concerned about mRNA vaccines, to be healthy. I don't want you to get infected and end up with respiratory distress and end up in the hospital. I don't want you to suffer. And that's really important. We shouldn't want this for each other. And explaining the science of how these mRNA vaccines are going to do that for that person and also potentially help the people around them are important points to make. So, with mRNA vaccines being a new technology, there are a lot of concerns about whether or not it is safe, or exactly even understanding exactly what it is. So let's go through a little refresher of what exactly is mRNA that we can use to explain in these kinds of conversations. Messenger RNA, or mRNA, is just a form of it's a step in gene expression. So it's transcribed from DNA. So we all have DNA in all of our cells, and the mRNA is transcribed by enzymes from DNA. And DNA is kept in the nucleus. RNA is kept in the cytoplasm outside the nucleus. So once the mRNA is transported outside of the nucleus, after being read from DNA, the ribosome, as I said earlier, is going to translate it into protein. mRNA, because of how this process works, is much less stable than DNA. So DNA is kind of like the repository in the library where all the information is kept. And then mRNA is like a transcript of notes that someone writes down from reading that repository and then taking it outside the library. So the point here is that mRNA is not going to be very stable and is going to degrade over time, which makes the components of the mRNA vaccines impermanent. And I think explaining to people that mRNA is something that is very essential to life and very common in all of our cells is also important. So these points of mRNA, the point that mRNA is constantly being produced in all of your cells, and that a single cell is going to have about 360,000 mRNA molecules at any given time, is important to express. The point being is nothing should be scary about mRNA. mRNA is a very common molecule in life, and we need it to survive, and our cells are always making it. So our cells are not seeing anything new or unusual when we get an mRNA vaccine. It's just another piece of code that our ribosomes are going to read to create whatever protein is being coded for. Along these lines of kind of demystifying or making mRNA vaccines less scary, I like to compare them to live attenuated vaccines, because live attenuated vaccines is something that has been around for a very, very long time and has a very, very long safety record. And live attenuated vaccines are essentially just a wild virus that has been grown or modified in a way that weakens it so that it becomes incapable of causing severe disease in humans, because it just isn't good at replicating in humans, or there's some other change made to it. And how these vaccines work is it introduces the whole live weakened virus into your body. And this virus is going to have a genome. It's going to have DNA, or it might have RNA. And it's going to enter cells and create what we call a limited infection. So it's going to infect some cells. It might reproduce a little bit, but it's not going to create a full-blown infection and capable of causing disease. And so it's going to function kind of similarly to mRNA vaccines, where during replication, your cells and your ribosomes are going to read the genetic material in the virus and produce proteins that your immune system is going to recognize and build an immune response towards. If anything, mRNA vaccines are arguably much safer, because instead of having an entire wild weakened virus introduced, it's just a piece of a virus. It's just, in the case of COVID mRNA vaccines, one mRNA and one protein, as opposed to a whole genome with lots of proteins. So I like this comparison to kind of demystify mRNA vaccines as something that is not totally new and unknown. So why mRNA vaccines? Why did we have mRNA vaccines in response to the COVID pandemic and not a live attenuated vaccine or a protein-based vaccine in the first year after the pandemic? Well, I already kind of went over one point, which is safety. It's like an mRNA, it's like an attenuated vaccine, but there's no pathogen. It's not infectious, it's non-integrating, and it's going to be a much simpler blueprint, essentially, for a vaccine. And the other big advantage that mRNA vaccines bring, and probably the most important reason that we had mRNA vaccines over another vaccine first, is the manufacturing capabilities, which offer speed and flexibility to mRNA vaccines that isn't offered to others. So we see in this timeline here just how quickly mRNA vaccines went to market, with the first COVID case in America being identified in first COVID cases being identified in January of 2020. And then we had the genome of the virus not long after. And once we had the genome, those sequences could be used to generate mRNA, or generate mRNA vaccines by companies like Pfizer or Moderna. And once you have the sequence and you just produce the mRNA and package it in a lipid nanoparticle with the technology for which had already been established, it was easy for these companies to hit the ground running and just put these products straight into preclinical trials or phase 1 trials and get the process rolling. So again, let's pause and recap. If the concern is that mRNA vaccine technology is too new and they don't think it's safe, we can explain that mRNA vaccines function really similarly to vaccines that have been around for decades, but function differently in ways that make them even safer. We can also explain that mRNA is not unnatural. Your cells constantly make it, and there's nothing really different about mRNA vaccines that would make it function any differently than your normal endogenous mRNAs. So now we're going to move into how mRNA vaccines are actually made, which is an important point to, again, demystify mRNA vaccines. So plasmids are pieces of circular DNA that are used in the manufacturing process of mRNA vaccines. I explained earlier that mRNA comes from DNA, it gets transcribed from DNA, so you have to have DNA in order to make mRNA. And this schematic here is just showing these circular pieces of DNA plasmids, which are separate from bacterial DNA in their normal setting. And biotech and biologists can use these plasmids that are usually found in bacteria to do a whole bunch of cool things. One of them, of course, is to take these plasmids for our own and modify their code so that they code for things like spike protein or other antigens we might want to code for. So this is just a basic schematic of how mRNA vaccines are made. You start with plasmid DNA, and then these plasmid DNAs are produced in specific settings, which are very highly regulated. And from once you have a lot of these plasmids, you can linearize them, which means you just introduce a single cut into the plasmid, which turns it from a circle to a long straight piece of DNA. That long straight piece of DNA gets moved into a what we call an in vitro transcription reaction, which just means that this DNA is getting read by an enzyme to produce mRNA in a system that is free of any cells. And once we have the mRNA, we can separate it from all of the other junk and package it into the lipid nanoparticle, vial it in a sterile container, finish it, and do all the other steps. And then you have your vaccine. So this is a little bit more complicated schematic of how an mRNA vaccine is made, just to kind of drive the point home of how much regulation I mentioned earlier goes into making these products. So every biologic is going to go through a process like this, where once you have your plasmid DNA, for example, the DNA is linearized in one step and then purified from a lot of the enzymes that were used in linearizing the DNA. And then it goes into the in vitro transcription reaction, and then there's another purification step. And then there's another manufacturing step and another purification step. And this goes on and on until you get your final product. There are a lot of steps, and each step has its own sets of testing and quality control components that eventually go into submission to the FDA before it gets approved. And all of these steps have to be demonstrated and pass certain very specific and strict criteria. And this is just a slide explaining the kinds of purification steps that might be used in a manufacturing process. A lot of these manufacturing processes involve steps that remove nucleic acids that are not RNA. So you can separate DNA out from RNA. You can separate out solvents or different ions. You can separate out the enzymes that are used. And all of it is just to introduce a pure and safe product at the end. And like I said earlier, each of these steps have very specific testing requirements. So at the end of the process, when you have your full drug product, there are going to be a lot of purity assays or tests that are required by the FDA for each batch in order for that batch to be released to the public. So in each batch, you have to do things like confirm the sequence. Is the mRNA sequence correct? Is it actually coding for spike protein? Was there some mistake somewhere that changed the sequence? That has to be confirmed. Is the RNA content making sense with the dose? Is the mRNA molecule made properly? Did the mRNA molecules get their required molecular components? Is it pure? Are there any residual DNAs, which is a big concern with some people today? Is there any is there any endotoxin or components left over from the bacteria that the plasmids were harvested from? All of these things and more are very strictly tested for. And it's important to make this clear to people who might be concerned about manufacturing processes. So again, let's pause and recap. mRNA vaccines being contaminated with DNA is a common concern that I have seen, and I have made videos about on my YouTube channel addressing in depth that it is out there. And this is very easily addressed just because of the fact that all batches of mRNA vaccines are very highly and strictly tested at multiple steps of manufacturing to ensure that residual contaminants like the DNA are being removed at each step. And attempts to show that contamination has been found have not really panned out. I've addressed these specific claims more in depth on my YouTube channel, but it's suffice to say here that these methods that people use to claim high contamination have not really been validated or qualified to show that these tests are measuring what they say they measure. Furthermore, which by the way is the standard that these manufacturers have to meet. And furthermore, I show in my videos on my channel that a lot of the math and methods they use are not appropriate for the claims that are being made. Essentially, what they're doing is way overestimating the amount of DNA they're detecting and claiming contamination, but that has not been the case in actual batch release data that has been reviewed by the FDA. So these claims are very easily addressed, but they can get quite complicated and nuanced. So this is just the base information that is needed to address them. So moving on to breakthroughs in mRNA vaccine technology. In 2023, the Nobel Prize in Physiology or Medicine was given to these two individuals, Dr. Drew Weissman on the left and Dr. Katlin Karikó on the right, two awesome scientists who spent many years developing technology necessary to make mRNA vaccines possible. And so let's go over just one of those breakthroughs here. Here's just a little timeline showing the amount of work and the amount of discoveries that were necessary to make mRNA vaccines a viable technology. And this, I think this is also important to explain when people think that mRNA vaccines are new and might be afraid of them because they think they're too new. The fact that they've been being developed for decades is an important point to bring up. This isn't just a technology that somehow got pushed through within a couple years. It took decades and a lot of hard work by a lot of brilliant scientists to make these basic science discoveries. And the one that we're going to focus in on here is this in 2005, which is the paper that was credited with winning Karikó and Weissman the Nobel Prize. So that's this paper here, Suppression of RNA Recognition by Toll-like Receptors, the Impact of Nucleoside Modification and the Evolutionary Origin of RNA. Essentially, what this paper is doing is it is explaining how our innate immune system, which includes things like toll-like receptors, which are these molecules here. The molecule is shown in this green and blue. And what it's doing is it's grabbing a piece of DNA. It's grabbing a nucleic acid. So these components of the innate immune system, these toll-like receptors, are trained to bind to foreign nucleic acids, foreign DNA and foreign RNA, because your immune system does not want foreign DNA or mRNA. That is a sign of a pathogen, so it's trained to recognize them. And so these toll-like receptors will grab onto this RNA and trigger downstream immune responses, which could be a problem if you're trying to use a nucleic acid like mRNA as a therapy or a vaccine. You want it to actually do a job. You want it to do something. You don't want it to just get recognized by toll-like receptors and essentially destroyed before it can do anything. So the introduction to this paper explains this background and asks essentially, why is it that the toll-like receptors are able to identify these foreign nucleic acids so easily, but it's able to differentiate them away from our own mRNAs and our own DNAs? And the answer came in the form of small modifications made to the nucleotide bases. So if you remember, nucleic acids like DNA and mRNA are composed of bases, which we often call letters. For DNA, it's A, T, C, and G. For RNA, the T is substituted with a U, and that's uridine, this molecule here. And this whole paper is essentially exploring this modification to uridine in mRNA molecules, which is called pseudouridine, which is just a change of this carbon here, changing from a carbon to a nitrogen. And so what Carrico and others noticed was that human mRNAs and human RNAs in general are much more modified with things like pseudouridine than their bacterial counterparts. So bacteria have far fewer modifications throughout their RNAs than humans do. And they also noticed that some RNA viruses have a lot of modifications, way more than humans. Maybe that's helping them go unnoticed by the innate immune system. So Carrico investigated what these modifications do to an innate immune response. And essentially what she did was she took mRNA molecules that have modifications and mRNA molecules that don't have modifications. And she used experiments to measure how much immune cells were responding to these different mRNA molecules. And I'll just draw your attention to this portion of this figure here, showing that modified or unmodified mRNAs produced a lot of this molecule here, which is an immune molecule, and modified mRNAs, which are pseudouridine is denoted by this trident-looking symbol, didn't produce as much of the immune molecule, and almost some modifications produce none. So this was a really important find. This modifications to mRNA could help the mRNA molecules not trigger this innate immune response. So essentially what we ended up with was something like this, where unmodified mRNA is triggering an innate immune response and a subsequent inflammatory response, and thus destroying the mRNA too quickly before it can actually do anything. Base modified mRNA is not triggering as much of an immune response. There's not as much inflammation, and the mRNA can actually stick around long enough for the ribosome to read it and produce a protein. So you can easily see that in an unmodified scenario, you don't get your protein, you don't get your foreign antigen, you don't get your immune memory response. Whereas in the modified scenario, you get your foreign protein, you get the immune response, and you get that immune memory. So this is just another paper showing that innate immune responses with modified mRNA still do produce an innate immune response. It's not like the modifications to the mRNA are completely ablating or completely avoiding an innate immune response. They are still producing an innate immune response. And that's just what this figure is showing here, that immune stimulated genes essentially are still being triggered when you have this modified mRNA. And this is all just to make the point that what you're essentially doing is you're tuning the innate immune response. Not directly, but indirectly by avoiding it. So again, the concept is if you have too much of an innate immune response with unmodified mRNA, you get no protein and no immune response. If you get too little innate immune response, then you don't get much inflammation at the site and you might not get as much immune cell recruitment. So mRNA vaccines have been developed over decades, not just with Carico's finding, but with several other scientists' findings to tune the immune response so that you still can get expression of the mRNA, you still get a protein, but you don't get too much of an immune response to not get that immune memory. And it's also tuned in a way that you get effective immune cell recruitment to the site of injection so that you get an even better immune memory response. But this has led to some confusion in the public space. Some people have heard about this Nobel Prize and thought that mRNA vaccines are actually directly affecting the immune response or dampening the innate immune system. And that's not really what's happening. It's just not triggering it. It's not affecting the innate immune system's ability to function. It's just not triggering it itself. So they don't suppress the immune system. They just avoid it in ways that allow for an effective immune memory response. And another claim or just concern about modified mRNAs in general is that the modifications made to the mRNA are dangerous or the dangers aren't known. Again, that goes into kind of this fear of new technologies. But the truth is that the modifications to mRNA are natural. They are found in nature. And modifications are also found in our own cells and in lots of other organisms. So there's nothing really unnatural about them. So there's no reason to think that the mRNA is going to function differently in our cells, any differently than the normal endogenous mRNA in our cells. So, but this goes into kind of a next big concern that I see just in general in the public. And it's this concern that mRNA vaccines are somehow causing cancer. And the truth is that mRNA vaccines don't cause cancer. But why is that? Why do we know that to be the case? It's important to understand a few basics about cancer before being able to talk about this. Cancer is really a disease of the genes. It's a disease of cells' DNA getting damaged enough so that the mechanisms that your cells have that allow them to work together break down, and rogue cells essentially just start expanding and multiplying in an uncontrolled way. And this DNA damage can be caused by things like carcinogens or mutagens. And typically for these things to cause cancer, you have to have multiple exposures over long periods of time, or you have to have persistence of the carcinogen. I mentioned earlier that the components of the mRNA vaccine are impermanent. The mRNA is going to go away, the protein is going to go away, and that is now shown here in this slide, where researchers looked at the protein that nucleoside modified mRNA can produce. And in this case, it's a protein that fluoresces, so they can measure it. And they injected an animal with this mRNA in multiple different ways. So that's what these abbreviations and different colored lines on the side mean. It's just intravenous or intraperitoneal or intramuscular. And intramuscular is the one that is relevant to vaccines. And we see that when an intramuscular injection is given, the protein produced by the nucleoside modified mRNA, it peaks at about just before day 1, and then it gradually declines over time to the point where it's undetectable. So this is just to show that these pharmacokinetic experiments have been done. We know that mRNA vaccines aren't going to stick around and continue to cause whatever issue might theoretically be responsible for any supposed cancers. It can't do that. It's not going to stick around and cause these things. But a big concern that a lot of people will pivot to is it's not the mRNA it's the DNA, it's the residual DNA from left over from the plasmid in the manufacturing process that might be causing cancers. And we know that that's not possible either. So why do we know that that's not possible? The reason is that, like I explained earlier, all biologics are made with, are going through these same kind of manufacturing quality control steps. And it's because all biologics are going to have DNA in the process at some point. Um, a, a vaccine might need cells to grow a virus at some point, and those cells have DNA. Um, Insulin needs to be produced by cells, and those cells have DNA. So all pharmacological biologics are going to have DNA. So manufacturers know how to test for it, manufacturers know how to remove it, and manufacturers or scientists have studied the potential harms of these residual DNAs in these products. So this paper here is doing just that. It's looking at the potential for DNA on its own to cause cancers. And what they essentially found is that DNA on its own cannot cause cancer unless it encodes oncogenes, or genes that code for proteins that can cause cancer. So what they did was they had they introduced DNA into cells in culture, and they also did this experiment with mice. But you see at this top row here, this DNA is just plain old DNA. It's got no special genes added. Conveniently, this particular piece of DNA is a plasmid, and it has a lot of the same components that people might be concerned about in the sequence of the plasmid used to manufacture mRNA vaccines. And when they introduce this DNA to the cells, there's no foci or bundles of cancer cells that they see. And an important point that's not shown in this image is that the amount of DNA being added in each case is about 1,000-fold. It's 1,000-fold more than the upper limit of DNA that is allowed in each batch of biologic. So again, no cancer with just plain old DNA. But when they add oncogenes to the DNA, you start to see cancer cells forming. And when they combine two oncogenes onto the same piece of DNA, you get a lot of cancerous cells. So it's these oncogenes that are exerting a cancerous effect in this residual DNA scenario. But the good thing about mRNA vaccines is that there are no oncogenes in the plasmid. There are no genes that code for proteins that can exert a cancerous effect. Um, so this is just a passage from this paper explaining just that, and driving home the point that DNA on its own, if without oncogenes, is going to have a potential of causing mutations or cancer-causing mutations at a rate that is less than the passage of time, essentially. So the point is, this topic has been studied. Residual DNA has a very, very low to non-existent risk when it comes to cancer. So the misconception that mRNA vaccines cause cancer is widespread online. We just went through a lot of the reasons why mRNA vaccines cannot cause cancer, why residual DNA cannot cause cancer when it does not contain oncogenes or is present in really trace amounts. But there's also the fact that cancer has not increased. Cancer rates have not increased in a way that would be attributable to mRNA. For this, you can go to just regular cancer reporting statistics websites like SEER, SEER, to see that the rates of cancers have not exploded after billions of doses of mRNA vaccines have gone out to the public, which you would expect if these mRNA vaccines had any potential to cause cancer. So that is just that's just how I would address that big concern that is unfortunately pretty common in certain online circles and public spaces. So to get to the end of this talk, we have to keep addressing myths with facts. Misinformation is going to keep coming. It's been around for a very, very long time. These kinds of fears about vaccines have been around for a very, very long time. A lot of them are not really new. They're just kind of repackaged in different ways to fit into the mRNA vaccine box. So it's going to keep coming, and we have to keep educating and addressing it so that people can continue to make informed choices. And there are going to be lots of specific claims that I can't address all in one talk. But in any case, I encourage you to just start with basics, go to the data that you do know, and follow up if you're having these conversations with people. And for specific claims that you might want to investigate between conversations, I recommend a source like vaxopedia.com or .org, sorry. It's curated by an MD and it's been around for many years and has lots of articles on lots of specific claims. And just finally, this is an image of a cartoon from over a century ago about smallpox vaccines. And the cartoon is basically showing that people are getting vaccinated with smallpox vaccine and growing little cows. And I include this just to show the fact that these kinds of concerns are not new. People, even before they knew what DNA was, had some concern that vaccines are going to affect um, some essence or some core part of their being in a way that is unnatural or scary. Um, and we know that smallpox vaccines helped eradicate one of the worst and deadliest viruses that we have dealt with as a human race. So despite these fears and misinformation, we did do that. So for those who might be feeling discouraged or um, a little hopeless in their efforts against misinformation, we have done these things despite misinformation in the past, and so we have to just keep continuing to do it in a new information landscape. So these fears are old, but they're still easily addressed with science and facts. And with that, I'm going to end the talk.
Thank you, Dr. Wilson. That was excellent. So with that, I do believe we've had a few questions come in. I'm going to just summarize here, if that's okay, Dr. Wilson. One is essentially just asking, how long is the normal vaccine approval process versus the emergency use authorization? And sort of as a follow-up, how can we ensure that there were checks and balances, like we're assured in doing such an expedited process?
Mhm. Yeah, that's another really common one that I didn't get to. But so normally, what happens in a vaccine approval process, you hear that it takes 10 years or 20 years to approve a vaccine. Uh, and whereas mRNA vaccines were obviously put through clinical trials and approved in about a year. So, why the discrepancy? And the answer is really just comes down to money and FDA wait queue times. So what happens normally is if there's a molecule or a vaccine that a company wants to develop, they will first have to allocate the funds or fundraise for it, and then put it through preclinical trials where they test it on animals. Um, and that is after developing and having a final product. They put it through animal testing, and then they collect those animal testing data and submit it to the FDA. And then they wait. They wait for the FDA to review it and approve it as a new molecular entity or whatever step comes next. And then they say, okay, you can move on to phase one. And then the company decides, do we want to move on to phase one? Okay, let's move on to phase one. Let's phase one trials with humans. Same thing. Collect the data, submit to the FDA, wait, get an answer. Do we want to move on to phase two? If the answer is yes, go to phase two, and again for phase three. And that takes a long time. It takes many years for that process to go through. And there's a lot of uncertainty in the terms of money. So a company is taking risk on a product. Um, they might not want to move forward very quickly with it. So with mRNA vaccines, money was taken care of. There was money, risk-free money provided by the government, the US government, to get these companies to try to produce a vaccine. We didn't know if a vaccine would be successful, but the government said, here's a bunch of money, try to produce a vaccine. And with FDA wait queue times, the companies didn't have to wait in line, right? Because normally when you submit for an FDA approval process, you have to you get put behind everybody else who submitted before you. But in this case, it was emergency, so the FDA had priority in reviewing COVID-related things, especially vaccines. So it doesn't actually take super long to do preclinical testing, to do a phase 1 trial, to do a phase 3 trial. It doesn't take very long to actually do those things and collect the data. A lot of the time that you hear about, a lot of the time involved in that 10-year timeline that you hear about is wrapped up in wait times and money issues. So those two things were not a factor in COVID vaccines. And the EUA process still included all of the steps. It still included preclinical steps. It still included a phase 1 trial. It still included a phase 3 trial. It still included all of those regular steps that would be involved in a regular approval process. It was just expedited. And I like to describe it as, you order a package on Amazon and you choose expedited prime shipping. You expect the product to get to you intact and the correct product to get to you intact and not broken. You don't expect a faulty product in an expedited process. It's just prioritized and expedited. So hopefully that helps.
Yeah, great analogy. Thank you for that. I think this is a great question to end on. It'll be a quick one. There's a comment that says, this presentation is fantastic. I'm learning so much. In our busy community vaccine clinic, we typically have just a couple of minutes to give a response about why mRNA vaccines are safe. In just a couple of sentences, without the benefit of graphs or data tables, how would you best respond to someone who's worried about the overall safety of an mRNA vaccine?
Mm, that is a good one.
Yeah, sorry to put you on the spot.
Well, I'll do my best, but I think if I just had a few minutes or a few sentences to explain to someone who doesn't know anything about mRNA vaccines, why they're safe, I would say that um, mRNA vaccines work very similarly to vaccines that we have studied for decades, and we know are safe. And in addition, we have had um, billions of doses go out all over the world. We've had thousands of scientists from multiple different labs, multiple different institutions, multiple different countries, all with different interests and backgrounds, all studying this mRNA vaccine, and they all agree that it's very safe. And if you think that a bunch of really competitive, nerdy scientists would all agree on something if it weren't true, then you haven't been to a Star Trek conference or convention. But something like that. I think I would emphasize that, we have a lot of knowledge about how these vaccines work. We have a lot of pre-existing research, and we also have a very global scientific community that has looked into this, studied it, and have not found anything to suggest that it is there are any risks that are outweighing the benefits.
Excellent. Well, thank you so much. And thank you again to everyone for joining us today. We look forward to seeing you again at our next webinar.
Thank you for watching the recording of New Technology, Old Fears, a walk through the science and myths of mRNA vaccines. This session was presented by Dr. Dan Wilson on November 20th, 2024. For those seeking CPE credit for watching this webinar, and for clarification, this is only for pharmacists and pharmacy technicians, please follow the details on the screen.